All right, I'd like to introduce our next speaker, Dr. Can Dincer from the FIT Freiburg Center for Interactive Materials and Bio-Inspired Technologies at the University of Freiburg in Germany. Dr. Dincer is currently a junior research group leader at FIT IMTech at the University of Freiburg. Their research focuses on disposable microsystems, the development of bioanalytical materials, sensors, microsystems in combination with data science and AI. So it'll be interesting to hear what Can has to say based on what we've just heard as well, for various applications, including diagnostics, especially point of care diagnostics and wearables. Since September 2019, he's an associate editor of the journal Biosensors and Bioelectronics. And in 2022, Dr. Dincer is also on the International Advisory Board of the Advanced Sensor Research Journal.
Thank you, Os, for this kind introduction. Welcome, everybody. Today, I'm going to speak about on-site therapeutic drug monitoring to individualize antimicrobial dosing. But before we start, I would like to raise two questions. So one of them is we heard also in the previous speech of Dr. Abdallah, but today I would like to also try from a different point of view to answer the question, does TDM-guided antibiotic therapy improve the patient outcome? And does it really help? And the second question is, what do biosensors promise us for therapeutic drug management? So starting with the first question, of course, in a usual scheme, we know that therapeutic drug monitoring is done in this way. So we took the sample, analyze it, interpret, and adjust the drug. And here we have, of course, chromatography and immunoassay-based methods. And what we are trying to give the clinicians the opportunity to measure more frequently by using sensors. What will be the advantage is, of course, the better patient outcome, like preventing side effects of those antibiotics which could be problematic. We believe that we could achieve a fast recovery, less mortality, and of course, this will lead also to reduce healthcare costs. And in the long term, it even could help to slow down the antimicrobial resistances if you really do the precise treatment. So to answer the first question, of course, does TDM help? So we found in 2022 the target trial study published, and so it was pretty interesting for us to look at a little bit near, since the conclusion of this study said a therapeutic drug monitoring guided therapy showed no beneficial effect in patients with sepsis and continuous infusion of piperacillin and tazobactam. And also with regard to mean SOFA values. And so they suggest it should be done larger studies. But is this really the case? So we would like to look at from another perspective by using data sciences. So we took this study with done in clinical in 13 clinical sites in Germany. And so at the end, where this trial has actually 124 non-TDM and 125 TDM patients in their study. And what we did is actually we took their complete data with these patients with about 200 features of each patient for each different day that they measured, and also try to create so-called patient cards. So these are actually mainly data frames that you can build it. And how we did this, actually, we did a very good collaboration with Dr. Atesh from Karlsruhe Institute of Technology, who helped us a lot with this models and also to understand and how to use them. So here we use this patient data, and the first thing that we need to do, because we had actually a lot of input, so the first thing was actually to some kind of sort the data, merge it, filter it, and so we get the secondary data where we then apply these patient cuts from day one to the day of their leaving the study. And actually, our goal was to use supervised and unsupervised models to see if we could see for the TDM a straight line from the sick to healthy status of the patient. So, and looking for the therapy success, we clearly see here, you can see the x-axis shows the TDM and or control patients who are away from the SOFA score 1. So we defined, because in our study we found out it would be better to take SOFA 1 as our healthy state. And what we see, actually, we looked the distance and how fast the patients are traveling to this SOFA score. And what we see, especially in the first 10 days, clearly, is the TDM patients, even some parts of it in a double speed, so they could they travel much faster. And here you can see also the day of the treatment. After the 10 days, it looks like that we have not anymore such a big difference. That was how we called actually how fast you get to this targeted SOFA score. And this showed us, actually, we use exactly the same data from the clinical study, but achieve a different results thanks to data sciences. And this shows us also that we need to use machine learning approaches to be able to look more clearly to this data. As I said, these are just received data that we get, and we would like to publish these hopefully also soon. And what we see is also regarding the SOFA statistic. Here you can see the last SOFA of the patients against the last day that they are leaving the study. And for example, 5 means that the patients leave the clinical study after the day 5. And here again, we see while the patients are leaving this study, they had the ones that had the TDM has a better SOFA score than the ones without any guided therapy. And the last point what we looked is also what we are interested is the recovery statistics. So here again, we had the cumulative sum of the number of the patients on one side, again, the days in time. So here we see also clearly that the cumulative benefit of the recovery is clearly much better than the ones that of the control and on the alive. So the patients who leave the study alive. What we see on the patients drop off the study because of their death, we see all the both cohort starts in the same, almost in the same conditions. We see that the slope of TDM is much smaller. And at the end, we see also less people are dead through this receiving the TDM therapy. And this shows us clearly that we need that the therapeutic drug monitoring really works on even clinical studies. And what we need, but now, and that's also the second part of my talk where I would like to highlight is, so here we had results everyday measurements, but we believe that if you can able to measure more frequently, even not continuously, maybe at the beginning, just maybe every hours for the beginning of the study, or a little bit faster, this will further improve the TDM practices and also its outcome. And therefore, the second part, I would like to show you our study working on a biosensor, which is capable of measuring antibiotics. And in this study, it was piperacillin tazobactam as well. We did a study with pigs where we treated them 50% of the normal dose, and also twice of the normal dose, and where we collected different samples of each animal, plasma, exudate, bread condensate, urine, saliva, and whole blood, and brought everything into our electrochemical sensor, and where we have an penicillin binding protein, which can and an ampicillin labeled ampicillin to measure beta-lactams. And so We published this at the beginning of last year, and one of first author of the study, Jeran, also won last year the Clay Award, which is one of the prestigious bioengineering awards in Germany. And what was our aim here? So we would like to measure beta-lactam. So our goal was, is it possible to have a solution like a glucose monitoring? So could we measure in whole blood the antibiotic concentration? Then the other option was, is it possible to use as a multi-sample measurement? So can we measure also drugs in these samples? And the last point was, is it possible to combine different drugs on the same system? So from the same sample. So what we saw actually for the overdose, normal dose, and underdose, we saw a clearly that we can measure antibiotics, and it makes also it's also fits to the values which are measured with the plasma values. So, and in the second part, we were more interested on the non-invasive samples. So because always to having a blood draw could be problematic. And one of the actually highlights of the study was we were able to measure in exhaled blood. Actually, we would not be able only to measure, but we could also see a clear clearance in the EBC samples, where you can see between plasma and exhaled breath condensate. So we can see nicely so that after the administration, the concentration increases, our signal is decreasing, and after a while, of course, it's clear, so that the normal signal is coming away. And this also dependent on the dosage that we are applying. We also tested other non-invasive samples like saliva and urine. And what we see here actually in those cases mainly is here we have the history of drug transportation. It's a cumulative information than an instantaneous feedback. So, and here our goal is of course to having a real-time feedback of the blood concentration of the antibiotics. In this study, we also showed that we can measure multiple antibiotics on the same chip. Here you can see one of the multiplex chip designs that we use, and for the time 0, so before the administration of the drug and just after the administration of the drug. And the same is also, we tested different samples on the same chip, so it's possible so that we can measure urine, saliva, EBC, or plasma together in the same system. Of course, as if you are creating a sensor, it's important to benchmark your sensor, and we did this by our single analyte sensors with the gold standard, with the HPLC. And here we can we see nicely that the t values, exponential values, are quite overlapping, which shows that our method is also delivers a good match with the benchmark systems. And we did then, of course, do the multiplex system and internal benchmarking from the single analyte to the multi-analyte of the chip, where we also nicely see that the results are overlapping. So in the final part, I would like to just talk about further possibilities. So so we show that we are able to measure antibiotics in different media and also within an hour, but our system could be even combine these together with the detection of, for example, bacteria or other pathogens. So this is actually a study which we did on COVID-19, where we combined the CRISPR-based nucleic acid detection of the COVID with the antibiotic monitoring. And the idea was kind of, and actually it could be also translated to the other pathogens like tuberculosis, for example. So once you are measured positively, and start to take the antibiotics, so you could do a personalized dosage adjustment so that you are always over an MIC value, over the MIC value, and after you are done, so you could be actually released. So, and here we are also further improving the sensor regarding its multiplexing capacity and also the readout devices. So we still have a benchtop device where we can go to clinics and do the measurements, but our goal is some kind of to make it more smaller, so that we can use, for example, a device which is a credit card format, which you can power up with a smartphone, and also do the whole analysis there. And this is some kind of also the ongoing work. With this, I'm end of my talk, and this is I would like to acknowledge, of course, all of my students. This is our faculty in Freiburg, and also the city behind, of course, it's coming. So, of course, I would like to acknowledge the PhD student, Jaren, who has done all the work together with her master's student, Regina, and also Jihan and Abdullah on the side of the side of the data sciences, and also Wilfried Weber and on the side of the synthetic biology parts, how to allowing us to measure. So I'm always finishing my talk like in this. So I believe that the science is about to share. So let's get connected if you are interested. And if you are asking yourself what social media got to do with the science, I have a talk in YouTube. Maybe I can convince you on that. Thanks again for this kind invitation, and thank you for your attention.
Thank you very much, Ken. That was very interesting and really built as a good follow-on talk from Alan's, and I think I've got a few notes that I've jotted down that I'd like to, speak to you and Alan about, and gather your thoughts, because this is getting to the stage where we are further going down studies that can really innovate how we do therapeutic drug monitoring, and also how we evaluate therapeutic drug